Hello and welcome. Today the Research Information Outreach team is joined by Dr. Sean Egan. Dr. Egan is a senior scientist at the Hospital for Sick Children in Toronto, and his main focus of study is breast cancer. Hello, Dr. Egan, and thank you for joining us today.
My pleasure.
So to start, tell us a little bit about how you first got into this field.
My interest in cancer would go back to undergraduate days. My interest in science in general and in biological science. I guess I would say I had a high school teacher, Mrs. Pudovic, that really encouraged interest in biology and asking questions, and that started me down the road. As an undergraduate, I had a fourth-year project supervisor, Herb Lejean, who was very interested in cancer genes and showed us some of the really exciting cutting-edge work that was happening in the 1980s. And then ultimately I did a PhD in the lab of Arnold Greenberg and Jim Wright. And they also, the door was always open for both of them. No matter how crazy my questions were, they really encouraged me to ask questions, even if it was unrelated to my project. And then I was very fortunate to get into the lab of Robert Weinberg, who discovered human cancer genes, and that was at MIT. And from his work, I then moved on to breast cancer research whenever I started my own lab. And that was really because breast cancer seemed like an unusual tumor. It seemed like the genes that were causing it were really related to developmental processes, a little bit different than some of the genetic causes of other cancers.
Yeah, that's very interesting. So over the course of your tenure, what is an obstacle that you find still challenges researchers today?
The field has changed quite a bit in terms of the size of it. There's been kind of a progressive expansion of the number of people working in science and society in general. And I think that's really driven, the innovation that comes from that has really driven our society forward. But we've kind of reached a point at which there's more of a contraction now, and so it's much more challenging to get funding. And in particular, a lot of the people that were unable to compete have now left science, and so there's a lot of very, very good scientists that are competing for a shrinking amount of money.
How do researchers go about acquiring funding in the first place?
So we write grants. We try to get grants from government agencies like the Canadian Institutes for Health Research, and but a lot of the funding in Canada comes from charitable organizations. For example, the Canadian Cancer Society Research Institute.
And once your research has been funded, how do you go about communicating your ideas to the larger community?
So it's primarily through publication and also presenting data at seminars.
I see.
As far as communicating with the general public, I guess that's not something that happens all that often. Sometimes publications are so impactful that the press picks them up and then discusses them, although that's usually associated with really a contraction of the complexity of the story. In other words, it's these stories that make it into the press, often the details of the story are completely lost and they all sound the same, which I think is one of the big challenges, is communicating to non-experts what the discoveries are and what the challenges are. Because all the stories end up sounding like, oh, we're 10 minutes away from the cure for cancer, and I don't think that's
true.
I don't think it's true. I don't think it takes advantage of the excitement created with the new discovery and the challenges that still lie ahead.
Of course, and it can be misleading to the public as well.
Absolutely.
Now, as an established breast cancer researcher, what are some of your biggest accomplishments?
So at each phase, of my career, I look back on something different. As a graduate student, I'll never forget that first eureka moment when I found something that nobody else knew for the first time, and that was I had a very nice paper. My supervisors put me on a fantastic project, which was really nice. And anyways, I found that some of the genes that are driving tumor formation, primary tumor formation, can also be involved in promoting the tumor's spread to other organs in the body. As a postdoc, I took some of the exciting work that was being done in fruit fly genetics and on some of the growth factor signaling pathways, and showed that it was also true in a cancer setting in mammalian cells. And then in my own lab, more recently, we've been trying to take advantage of some of the human genomics the human sequencing efforts that are looking at the sequence changes that have occurred in human breast cancer, and then trying to make mice that model specific kinds of breast cancer. And then I've just learned from a colleague here at SickKids, Michael Taylor, how to do something called a Sleeping Beauty screen, and that's really changed everything we're doing in the lab, because it's given us insights into the whole genetic program by which cancer cells spread from the breast to the lungs or liver or other parts of the body. And I guess I would say that what we're doing right now is, for me, the most exciting stuff. You phrased the question asking about my successes, and it's important to note that this is a continuum. So many of the papers that come out of my lab involve 15 authors. And most important of those is the first author, or sometimes the first two authors working together, have done 95% of the work, and they're often a trainee. So, it's not really me, it's we.
So life must have been very different for you back then as a young grad student. How have sequencing technologies changed the daily activities in the lab?
So as you know, most of the daily activities involve pipetting colorless liquids from one tube to another, and using machines that spin, shake, cool, or heat. So the lab work looks kind of the same, but the power of the lab work, because of the details and the specific machines you're using, mean that instead of looking at the effect of one gene at a time, or instead of studying one gene at a time, you now study the whole genome, 20,000, 30,000 genes at a time. So the amount of data that comes out of your experiments is logs more than it was before. One of the major misconceptions, and I think this comes from various places, is that we have a lot of clues now, and it's time to really move from basic science into translational science exclusively. We should always have, I think, the full spectrum. The problem with cutting off basic research funding is that that's the foundation for where completely new ideas come from. And it's a bit like if the Blue Jays got rid of all of their, AAA and AA and single A teams, you would quickly run out of new players and the team would collapse pretty quickly. The use of model organisms like worms and flies, and we use mice to study cancer, has just completely transformed the way that we think about cancer. More recently, people were studying bacterial immunity, how bacterial cells defend themselves from foreign DNA coming from bacterial viruses and things like that. And they discovered something called the CRISPR system. That wasn't very long ago, and people are already talking about using CRISPRs to modify the human genome to treat people that have Duchenne's muscular dystrophy, or to try to figure out a way to actually repair the gene. So if we cut that off at its source, we'll run out of ideas very fast.
Yeah, and none of these studies set out with the goal of discovering something.
Absolutely not. They were discovering something fundamental. Someone just asked the question, how does a bacteria defend itself?
Exactly. So
it was just curiosity-based research.
Finally, what does the future of cancer research look like to you, and where do you think it's moving towards?
We take a certain angle on it, and so I'm really excited about a lot of the mouse models we've made for breast cancer, trying to model the different forms of breast cancer, model inflammatory breast cancer, or some of the aggressive luminal type breast cancers, basal breast cancer. So, and then using those models to find new therapies, using robotic screens on cancer cells from the mouse, using those models in co-clinical trials to try to figure out if the cancer becomes resistant to the therapy, how did it do that, and can you come up with a second treatment? But in a more general sense, like, I wouldn't want all the funding to go to only the kind of research we do, that there's some very exciting work being done by other people in immunotherapy and areas that I don't fully understand, but clearly are pushing forward with really novel therapeutic approaches.
Well, thank you very much for your time today, Dr. Egan. I think we've all learned a little bit about what happens behind the lab coat. And thank you all for joining us today. I hope that you have enjoyed today's session. And please visit us next time for more upcoming Riot videos. Thank you.